of the research prize of the Swiss Society of Cardiology. This time it's somebody from Bern. It's Dr. Rexhaj Emrush. He trained in Lausanne and in Bern and is now very much interested in vascular disease and hypertension there. And the prize award goes to a paper published in the Journal of Clinical Investigation, a very prestigious journal. And the title is Mice Generated by In Vitro Fertilization Exhibit Vascular Dysfunction and Shortened Lifespan. Dr. Rexhaj, welcome.
Dear Chairman, ladies and gentlemen, thank you very much. First, I would like to say thank you to the Swiss Society of Cardiology for giving me this prize and the opportunity to present you here my work. Well, there is increasing evidence that pathological events during the fetal or perinatal period may have important consequences later in life. For example, data from our group shown in humans that assisted reproductive technologies or preeclampsia cause vascular dysfunction in the offspring. This vascular dysfunction may put these children at an increasing risk for premature cardiovascular morbidity and mortality. Because it is difficult to study the mechanism in young, apparently healthy children, we generated a mouse model and started to analyze vascular function, arterial blood pressure, and the mechanism underpinning vascular dysfunction. Let's see now the results. This slide shows mesenteric artery responsiveness on the left side and arterial blood pressure on the right side in mice generated by ART and control mice. In ART mice, shown here by the red line, the mesenteric artery vasodilation evoked by increasing doses of acetylcholine was impaired as compared to the control shown here by the blue line. This defective vasodilation in ART mice was caused by endothelial dysfunction because, as you can see here on the graph on the middle, the endothelium-independent mesenteric artery vasodilation evoked here by increasing doses of an nitric oxide donor, that is sodium nitroprusside, was comparable on the two groups. The graph on the right side shows invasive measurement of blood pressure in ART and control mice. ART mice are shown in red and control mice in blue. As you can see, blood pressure was higher in ART mice compared to control. Thus, so far, we have demonstrated that, as in humans, ART induces vascular dysfunction and arterial hypertension in mice too. And most interestingly, we found that ART mice transmit the vascular dysfunction to their offspring. This slide shows again mesenteric artery responsiveness in ART mice and their offspring. You can see here in the green line that mesenteric artery vasodilation in offspring of ART was comparable to their father. For comparison, data from control with the blue line are plotted again. This transmission to the next generation suggests the possibility of an epigenetic mechanism underpinning vascular dysfunction in ART mice. In line with this hypothesis, it is now well established that during the early embryonic period, here on the left side, there's a vulnerable period with high epigenetic activity that is associated with fetal programming of cardiovascular disease. DNA methylation is an important epigenetic mechanism that affects gene function and fulfills the criterion of heritability. The manipulation related to ART could induce DNA hypermethylation, shown here. that in turn can affect the gene function and induce vascular dysfunction. To address this issue, we first examined whether ART-induced dysmethylation of a gene known to be involved in vascular regulation, namely the endothelial nitric oxide synthase gene. And as you can see on this slide, we found that DNA methylation of the promoter of the ENOS gene was increased in arterial tissue of ART mice, shown in red, as compared to the control mice, shown in blue. It is known that promoter DNA methylation regulate ENOS expression in cells in vitro. Naturally, the next step was to assess if ENOS expression was affected. And here I show you the ENOS expression in the carotid artery. As you can see, the ENOS expression was decreased in our mice compared to the control animals. Decreased ENOS expression was of utmost important because, as I will show you on the next slide, the plasma nitric oxide concentration was markedly decreased in ART mice compared to the control animals. Well, what could be the long-term consequences of this problem? Here I show you Kaplan-Meier survival curve of ART and control mice fed with normal chow or high-fat diet. ART fed with high-fat diet are shown in the red line, control with the high-fat diet with the blue line, ART with normal chow with the orange line, and control normal chow with the green one. As you can see here, whereas high-fat diet had no effect on the controls, the mortality in ART mice fed with high-fat diet was markedly increased. And in conclusion, I can say that this study shows that ART per se induces vascular dysfunction. This vascular dysfunction is induced by epigenetic mechanism involving at least decreased NO bioavailability, and the lifespan in ART mice challenged with a high-fat diet is roughly 25% shortened. And at the end, I will say thank you to all my group that helped me to realize this study. And thank you for your attention.